Hello everyone, my name is Sarah. Today I'll talking about effect of cell age on the lag phase of bacteria. First, I will introduce what is the lag phase. Then I'll describe my aims, hypotheses, methods, results, and then give the overall conclusion. I would like to introduce my topic by talking about what is the bacteria lag phase. What factor influence the duration of the lag phase, the bacteria that are being studied, and why I chose this topic. Lag phase is the period during which bacteria rejuvenate and adapt in their environment before growing exponentially. During the lag phase, individual bacteria adapt themselves to the environment in order to attain optimal growth rate. This graph shows the typical bacterial growth curve. Comparatively, few studies have explored the physiological aspect of lag phase. However, it is known to be affected by several factors, including temperature, pH, environmental stress, nutrient availability, inoculum size, and period cell history. In the study by Thomas Listeria, aging is a part of the life history of bacteria. Like all live bacteria age, and as they grow old, they slowly accumulate damage, and that at some point they may stop growing. The division of E. coli could describe the reason of cell aging, which is one effect factor on the bacteria lag phase. The picture shows the life cycle of E. coli. During cell division, two new pores are formed, one in each of the progeny cell. New pores shown in blue. The other end of this cell will form during a period division, old pore shown in red. This process of forming pores is called cytokinesis. This representation of life cycle has been serialized by Listeria to describe cell aging. Since during cytokinesis, one daughter cell will inherit a pore that is older than the one inherited by its sibling. By this reason, the old pore cell of E. coli display a progressive increase in their generation time, as indicated by cell length based on microscope observation. This asymmetric response could also contribute the heterogeneity in the cell population. As time goes by, more heterogeneity emerges, and it could be cell age. There are other factors that can affect the bacterial lag phase, such as the inoculum size. The internal number of active cells in the population, how quickly visible growth can be observed. That means a large number of active cells would quickly reach a level of visible growth than a small number. Thus, when inoculum size larger, the lag phase is shorter. As also temperature increase within in the normal physical range of the cell lag phase is also shorter, probably because the biochemical reactions are more rapid. The bacterial lag phase also depends on environmental conditions, since growth rate is roughly proportional to lag phase duration. Why I choose these two bacteria? Because these two bacteria are in different phyla. In terms of growth properties, these two bacteria are not very different, but the E. coli has a narrow range of growth temperature and pH. Another reason is that E. coli and Listeria can cause safety issues in many foods. Thus, we want to be able to accurately know how these bacteria grow and behavior in food products and the food safety chain. This experiment aims the period cell history affect duration of the lag phase in E. coli. From this aim, we can assume that different age of Listeria and E. coli have a different lag phase, and older bacteria have a longer lag phase. This graph shows how I did this experiment. A slope of Listeria monocytogenes FW0400025 and E. coli M23 was transferred into 100 ml triptosoyabros plus 0.6% yeast extra, and they inoculate at 37 degrees for overnight. This stage was for preparing the inoculum. Each 9 lots of 100 ml inoculum was transferred into new fresh broth, and each lot then inoculated at 37 degrees for different days. Inoculation days was 1, 1.5, 2, 3, 4, 5, 7, 14, and 21 days. Samples were collected several times on these days. Listeria diluted into 10-3, and E. coli diluted into 10-4. Different stage population of these two bacteria were collected together. The experiments were replicated in order to make sure that inconsistent results could be checked. Value on the graph are average, while the error bars are standard deviations. The lag phase was calculated at a point when a significant increase in population occurred. Here, the lag phase was considered to be finished after 140 minutes. This graph shows the population of Listeria of different ages that were measured for lag phase. We can see at 1 and 1.5 days, the lag phase time is 80 minutes. In 2, 3, 4, 5, and 7 days, the lag phase ranged from 100 to 120 minutes. However, at 14 days, the lag phase was 210 minutes. At 21 days, the lag phase was 330 minutes. Comparing all of these inoculation days, the lag phase of 14 and 21 days was longer than that of the other cell age. From this graph, we can say that the 1 and 1.5 inoculation days, the cell age is inoculum is young. At 2 to 7 inoculation days, the inoculum appears to have a median lag phase. While at the 14 and 21 days, the effect of cell age are very evident by increasing lag phase duration by a factor of 3 to 4. This graph is the population of E. coli in different stages. In 1, 1.5, 2, 3, and 4 inoculation days, the lag phase time was ranged from 40 to 60 minutes. The lag phase of 5, 7, 14, and 21 inoculation days were 80, 120, 180, and 300 minutes respectively. Comparing these all inoculation days, The longer lag phase were 7, 14, and 21 days. Contrast with Listeria, the young age was 1.5, 2, 3, and 4 inoculation days, and the old age was 7, 14, and 21 inoculation days. The lag phase of E. coli was shorter than Listeria in all of the inoculation days. However, the effect of cell age at 14 and 21 days increased the duration of the lag phase a lot, by 4 to 5 times. Perhaps E. coli age more quickly than Listeria since it grows quicker. The results showed as the population age lag phase linearly increased for both of these two bacteria. The two strains lag phase duration were very similar across the spectrum of young to old cells. The slope for E. coli is slightly deeper, which could indicate a slightly more rapid aging, and that's a greater effect on lag phase duration. Other bacteria that grow even more quickly and more slowly could need to be tested to see if this is the case. While lag phase is increased with cell age, the number of genes differently expressed for smaller in old cells than in young cells. Relatively few genes were up and down regulated in both cultures. Transcription of genes related to different intracellular activity in young and old cell. Young cells rapidly express genes during lag phase and don't express stress protect gene, suggests they don't need to repair damage before growth. However, old cells seem to express some stress responsive gene, and that's probably that repairing and adapting at the same time. Thus, they take longer to live a lag phase. From this experiment, the result confirmed that cell age is an important factor affecting lag phase duration, and may reflect physiological change with the population as well as increasing degree of cell component damage, that therefore require more work to achieve optimal growth rate.